as a process engineer in the chemical development department at the AstraZeneca MacLeod site. So our role is scaling up processes from the lab to the plant, whether that's our internal manufacturing assets or the external manufacturing assets, and checking that all the unit operations that we use in the lab are going to work in the pilot plant and production scale plants. So cloth blinding can explain the performance seen, although it was more extreme than seen in the lab. And was that down to not being able to replicate that properly in the lab, or was it down to the fact that we only studied 5 cycles? An increase in cake resistance from the sort of the good drops to the bad drops also explains it, but crucially, we didn't detect any of the undesired form that is known to cause the slowdown in filtration and the increase in the filtration resistance. So it definitely wasn't down to the presence of the undesired form. Could it be down to changing the particle size distribution? Again, that was measured and it was found to be similar from batch to batch. So unless it was quite a subtle effect, then we don't think it was down to the characteristics of the cake itself, although it is something that we'll be looking out for on the next campaign. So going with the blinding theory, then centrifuge cloth washes will be implemented on the next campaign. If those washes don't help, then a new cloth will be selected, going back to the cloth vendors to see if there's something they'll suggest for this particular morphology of crystal that does seem to be quite prone to blinding, to see if there's something that's designed to be a bit more robust to blinding. And then the Dynochem centrifuge, the Dynochem centrifuge model we found was helpful to use with these mass traces to try and see what was going on, what variables would change to give behaviour more similar or less similar to what was seen. So we found that very helpful. The question that we're now getting asked from the chemists and the project managers is, can we scale up directly from the lab to a centrifuge on the plant? And the answer is kind of partially yes, I would say. So from the filtration cake properties and equipment dimensions, then we can estimate cycle time, we can optimize drop size and feed time, we can compare the cake and liquid height, which means we can compare the pressure drop. It enables comparison between 2 different types of equipment and what the implications would be if we try and match one parameter on the equipment, say the G-force, then we might end up with slightly different heights of liquid above the cake, for example. However, I would say a lab centrifuge or centrifuge trials would be required to look at things like wash efficiency, to look at deliquoring, and to look at the impact on particle properties. So it depends what it is we're looking for. If it's around the kind of how long will my filtration take, absolutely we can do that from the simple lab filtration and the model. If it's what are you going to do to my particles, or are you sure that you're going to wash out this impurity, then that's where we might need to do some lab scale experimentation with the lab scale centrifuge as well. So our proposed workflow is making good use of our lab scale filtration unit to calculate our filtration crate properties, alpha, the cloth resistance and the compressibility, using the model to work out what we should have as our drop size and our feed time and our spin speed and so on, and then any further comparisons that we need to do between different sizes or different types of centrifuges we can also do with the model. We would use the lab centrifuge for targeted experiments, and that's where things like wash efficiency, particle size, or deliquoring is important. So it's going to be a combination of the experimental and modelling approach that we'll go on to in the future.